of bioinformatics, and I just want to see, does anyone know what an informatic is? Can you please type it in the chat? All right, so that's fine. So, oh wait, let me check. So an informatic is basically a data system that we use to access different data points, and we can insert in data. So I'll show you guys the definition of bioinformatics.
Hello and welcome to the Ohm Genomics Show. I'm Maria Nadestad. Today I'm going to tell you a little bit about how I define bioinformatics. So here's what I tell non-scientists that I do. I use computers to analyze biological data. So that usually helps them understand. But just to be sure what the definition of bioinformatics was, I went to the Almighty Wikipedia and to check that I wasn't crazy for this video. And it defines bioinformatics as an interdisciplinary field that develops methods and software tools for understanding biological data. So I think that's a pretty close definition to what I had. And people in bioinformatics also like to make a really big deal about that Venn diagram where biology meets computer science meets mathematics or statistics. And this actually looks a lot like the Venn diagram that the data scientists make, where it's computer science meets statistics meets domain knowledge or domain expertise of some kind. And here, of course, domain knowledge and domain expertise is biology. So maybe if we want to impress our friends and improve our earning potential, then we should start calling ourselves data scientists or biological data scientists. But depending on what we do exactly, the title could also be bioinformatics software engineer or something completely different. It really depends a lot on what you do. So I've heard multiple people debate the difference between bioinformatics and computational biology, and they really don't agree between different people on what the difference between these two fields is. And I think a more useful distinction is these three major approaches to doing bioinformatics research, which are number one, data analysis. This could mean going from raw data to cleaning up the data, and then to doing some statistical and visual interpretations of the results. But it's all focused on taking a data set from the raw data to some kind of an answer at the end. The second one is bioinformatics software development. This is developing software to do bioinformatics analyses, and these are big enough software products to publish as independent methods papers and to be used by other scientists in the community of the field that you're studying. The third one is modeling, and this is modeling simulations, generally making equations to represent biological systems. So these are three different categories that I've had some experience with for all of them. And in my experience, seeing the broader field and who's doing these different kinds of approaches to bioinformatics, I would say that data analysis is the most natural starting point for biologists. It's what I started with. And it involves the most domain expertise because it specifically involves interpreting data. The ability to detect oddities and interesting patterns in the data can also heavily depend on your knowledge of the biological system that the data comes from. And so it's really important to have that biological domain expertise in order to do the data analysis. So for the second one, bioinformatics software development, I've experienced this approach as something computer scientists naturally take on. They can also do data analysis, but most computer scientists will have a hard time resisting building some kind of a real software product along the way. And the software that is developed can take many forms from command line tools to web applications, also something that I've done. And maybe I'll talk about a little bit of what I have done in my own story and how I've moved through these fields. The third one, modeling, I've seen as very fashionable with physicists and mathematicians. Get it? Modeling, fashionable. You can tell their work apart by the fact that it's full of equations and sometimes written in LaTeX. So I'll give you an example of each of these three things based on my own experiences and things that I've worked on, because I do have a bit of experience in all of them. And so the first one, data analysis, as I was coming out of college, I had mostly wet lab experience. And my first major project in my PhD, and which became most of my thesis, was this analysis of some PacBio sequencing data in a breast cancer cell line called SKBR3. And what I did there was we started out with a lot of sequencing data. So I had to use some pipelines like aligners and variant callers in order to analyze that data. So you go through the pipelines and then you end up maybe making some plots and things like that at the end. And so that was what we did. Now, for the second one, this goes very well. We ended up needing to build our own bioinformatics tools because the ones that were made for next generation sequencing, which we had been using for some of the steps, were actually not sufficient for dealing with long read sequencing data and getting all the full information content out of that. So we ended up building some of our own tools. And I built some of those, and my colleagues built some other ones that were more optimized in C and so on for making it super fast to run through a lot of data, and that's really important. What I had done was some more of the interpretation, coming from that biology standpoint from the beginning. I was doing some algorithms that would help us to interpret the data as well and kind of consolidate it and bring different data types together. And also, I built visualization tools and kind of analysis coupled with visualization tools. Split Threader is a really great example of that, where you do some work on the back end, in Python or R, and then you take that and you show the results of that to the user in a nice visual interface. So that's an example of a web application that actually has command line tools on the back side, but then also shows you it in a visual interface. And for the third one, modeling. I haven't done this in my PhD so much, but in college I had an undergraduate research project with a math professor and another student where we were doing a network to represent the students on campus, and we would see how the flu could spread around the network based on who knew who else in the network. So like the nodes or students, you'd use like probabilities for saying whether the flu is going to transition be spread from one person to another in this network. And so kind of simulating that spread on the network and also predicting what would happen in terms of using probability distributions, but also doing simulations and seeing if they fit together. So that kind of encompasses, I think, nicely an example of what that modeling approach to bioinformatics is. So now I want to hear from you.
All right, so bioinformatics, as you can see, it's a very vague, it's a big subject, and there are so many crossovers. The definition is the science of collecting and analyzing complex biological data, such as genetic codes. So this can be used like a lot. So I just went on the internet and I looked up bioinformatics, and all of these came up, and I think even more. But so right now, what I'm gonna use is we're gonna talk about biodiversity informatics and then biological ontologies. And obviously we can't get through all of these. So we're only going to go through some important steps in biodiversity, in bioinformatics. Okay, so biodiversity informatics. So what do you guys, so what do you guys think this means? So you guys know what informatics are, and biodiversity informatics, you put them together. What do you guys think? Can you guys type in the chat? Yeah, Olivia, that's a step of biodiversity. Well, that's actually a step of the next slides we're going to talk about. But yeah, that's correct. So we can analyze the data on species interaction. So basically, what the term X means is, so biodiversity is the variety of life, and informatics is a science practice or process of collecting, organizing, storing, analyzing, preserving, retrieving, and governing data records and relevant to an organization. So this is for biodiversity. So basically, the definition is the collection and analysis of biodiversity data. So basically, what it is handling of any biodiversity information. So it records different specimen that we find in order to like map out the like if they're invasive or endangered or not. So the first process in biodiversity informatics is we first have to get the species, right? So we have to collect the specimen, and most commonly it's like a plant or something, like I don't know, like some tree. So what people do are, so basically uh, we collect the specimen, and then you can just, there are people who collect the specimen and then they can just all right, so my screen kind of went blank for a couple of seconds there, so I'm sorry about that. So here we can collect, and then so we can distribute specimen by so we can distribute the specimen by, so there are people who collect there are people who collect these specimen and then they distribute it to labs and such. And then sometimes it's kind of like, I don't know how to say, unethical, because like sometimes you collect some, I don't know. So last I had a rodent, I had a squirrel in here. So basically what happens is you have to collect them and then you have to preserve them. So that means that you have to humanely get rid of their consciousness. So it's not a very nice and fun thing to do, but it's extremely important in order to map out the ecosystem and stuff. So what basically happens is we collect and then we humanely kill them by using CO2 tanks. And then in Russia, they have a statue of lab mice. These are obviously lab mice, not the wild species, but same principle. And then I looked up something in America, and then you can't, there's America honors lab rats here, and it just, there's nothing. It's just the Russians honor the lab rats, but not America, which is kind of sad. And then, so why is this important? Like with all of this, it's kind of sad how we have to do this stuff, but it's extremely important. Like the it's extremely important because we have to map out the climate, like what the organism has a impact on the environment, or if it's like invasive, if it's invading the area, or if it's like endangered. We can help a lot more if we just have to take a couple samples. And here's a paper I found regarding insects' response to climate change. So this is also a use of biodiversity informatics. We can find out how the insect population is adapting to climate change, which is very important because climate change, we kind of cause that. We're causing it right now. And we need to find how it's affecting these animals. So the second process or step is to prepare the specimen. So see, usually it's a plant, like a plant invasive species or endangered or such, for scientific research. And then we digitize that information in order to store it in our database. So right now, we live in the 21st century, which means that we have access to all this electronic information. It's amazing. So in the past, the ecologists made up data sheets and they would like hand write them. And that's not a very efficient way to make and share your data. Like for a nurse, this is a nurse's office. So a nurse, you can make like post-it notes to show which patient you're gonna see and when you're gonna see the patient. But if you're gonna have data, if you're gonna have data like stored, it's probably better to have it uploaded and digitized. And then what we do is we standardize the information. So we can change the format, load it to different database systems and different languages. And this is extremely important because we want to have as many people access to it because this is important data. And then we publish it. That's pretty self-explanatory. And then before I move on, do you guys know some other possible purposes of biodiversity informatics? So yeah, you guys are right. So like in agriculture, bee pollination, you can map out the bees probably, and then Olivia, the data can be used for connecting problems. Yeah, that's true. Like if you have a problem regarding like the ecosystem or the environment with the specimen that you find, then it's very important to share it. And then more info on the animal food chain. Yeah, you can find out more about the animals and its role inside the environment and predicting. That's a good point, Olivia, predicting the future. So basically, what you can do with this data is you can predict what will happen next. So say like you have an invasive species migrating over, let's say to, I don't know, Nevada or something. So then you see Nevada is gonna, 2 years later, you see this whole thing, like, let's say they're tumbleweeds rolling all across the place. So you want to be able to spot that and see how that'll happen. All right. So you guys answered quite a bit. That was really good. So you can basically tell if the species is endangered. That's very important because, I mean, humans are kind of the problem right now. And I think it's a good idea to kind of delay the problems or try to stop our problems because what we're doing basically is we're changing the environment so much that we're killing so many animals and insects, changing the environment that they can't adapt fast enough so we can find the endangered species. And then invasive species. This is important because if you have an invasive species, it can take over, like Nevada, I said. Like the tumbleweeds, they just spread so fast, and then it'll like all the natural vegetation there is kind of, it's going to die most likely, and we want to preserve that. And then we can tell if it's migratory, so if it's like migrating, and then if it has an impact on climate change. So basically what we can do with that is we can just catch humans and then, we got them. We just have to upload it into the database. And then we can find the genetics of the specimen that we found, which is important in evolutionary biology and such. And then evolutionary standpoints. We can find our ancestors this way. Here's our closest living ancestor, and then these guys are extinct. And then the ecology. So basically, we can find the way they interact with the environment, whether they benefit the environment, or they like us, they kind of destroy the environment. So it's very useful to use biodiversity informatics because like I said, if we can prevent and store this data, like this, the prediction map, we can prevent these tumbleweeds. This is in Nevada. This is more like Illinois. But so we can stop it from invading Illinois and possibly take preventive measures, put out maps so then we can show our data. And then it'll help our environment if we keep this data. I know it's kind of sad for some of the collections that we have to do, but it's well worth it, I have to say, because it saves the environment more. And overall, we can use like UVAs to like map out the system. We can just take like pictures, and then that'll show like quite a bit of the invasive species and such, or the endangered species. All right, before I move on to this presentation, I want to ask you guys. So, biodiversity informatics. We went over quite a bit, and how do you think we can improve this technology we currently? Like let's say we can have more imaging in the sky to find these species. Yeah, finding more ethical ways to collect data will probably be very good, because it's like science is it's for the good of we'll say for the good of the ecosystem or humans, but the specimen didn't really consent to like being tested on. but for the greater good, as Aaron says, yes, for the greater good. But there are some, like, we can use imaging systems, and I think animals are less likely to be collected compared to plants, so it's not as bad, but it's Harry Potter meets. Yes. All right. Any more suggestions? Making it easier. Improve the accuracy of data and making it easier to input data. Yes, these are very good points. Inputting data can be a challenge for ecologists, but I don't really know how much data and how they do it per se. But improve the accuracy of data, we can probably do that using better imaging systems. Like right now they use UVAs, which are like airplanes in the sky with no pilot to take pictures. So in the future, we'll probably have better imaging systems, which will give us like more accurate data probably. And then we can predict with increased accuracy of like what's going to happen with the tumbleweeds. Are they going to happen? Are they going to destroy the vegetation in like 2 years or 1.5 years or such? Yeah, minimize the invasiveness of collecting data. Like it's a bit invasive for some things, and yeah, that's something to be taken into consideration. So then we can like have less of an impact on the environment. I mean, we don't really have that big an impact, but I honestly don't really like it, but of course it is, as Aaron said, for the greater good. But yeah, if we can minimize the invasiveness and find the best data, then that would be pretty good. All right, now let's move on to biological ontologies and data integration. So first of all, do you guys know what an ontology is? Type it in the chat if you do. Oh, philosophical, that's so funny. So there's another definition for ontologies, which is like the metaphysics of something, but that's not a biological ontology, or it's not an ontology I'm talking about, but that's really funny. So concept that shows relations between stuff. Yes, that's correct. So yeah, that's funny because I looked it up and it was exactly what you're talking about, Olivia. So an ontology is a formal naming and definition of types, properties, and interrelationships between particular entities in a domain. So what that basically means is that we can find like definitions and relationships between like classes, which we'll talk about later. So classes and our data. So how is it used? So basically what it is, it's used for community reference. So like you have to search, integration, and exchange of biological data. So this is very useful in like finding like data or like finding a reference point, maybe even a definition of something. So an example, so here are the examples of biological ontologies. So we have controlled vocabularies, which is like a thesaurus, like this guy right here. So, and taxonomies, I'll go on about that later. And thesauri, which I already said. And then how are ontologies used? So this is where the confusing part comes. So we have classes, relation, and attribute. So I'll show you guys a video about this because, to be honest, this can explain much better than I can. So I will link the audio. Okay, so can you guys still see my screen?
Welcome. In this session, we will focus. So there are three things that we define in the ontology: types, properties, and relationships. Let's take a look at each of those. In Party, types are called classes. A class is a way to define a set of concepts that share certain properties of relationships. This means that once you have a class, you can say that all instances of that class are allowed to have a certain attribute, or the two classes can have a specific type of relationship. Classes can also have subclasses, or be subclasses of other classes. If a class is a subclass of another class, this means that all instances have to be members of the superclass before they can become members of the subclass. It is important to understand that in the ontology you define the class and which attributes and relationships classes are allowed to have, but you do not actually define the attribute value itself. In the ontology, all you do is describe how things can be, but not how they actually are. In other words, in an ontology, you define which information you are allowed to associate with certain concepts, but you do not specify the actual information itself. Let's take a look at an example. We could say that we have an ontology we want to use to describe cocktails and everything you need to make cocktails. You could, for example, define a class called alcoholic beverage. For that class, you could define certain attributes and relations, and we will get to that in a minute. You could also define a class called beverage. Now, an alcoholic beverage is a special kind of beverage. This means that every alcoholic beverage is also a beverage. In an ontology, we would model that by saying that alcoholic beverage is a subclass of beverage. In order to be a member of the class alcoholic beverage, a concept also has to be a member of the class beverage. Let's go to the next thing we define in an ontology: properties. Properties are called attributes in Pool Party. Every class can have attributes. This means that once you have defined a class, you can say that all concepts in that class are allowed to have a certain attribute. Attributes are a way to associate all kinds of information with concepts in your taxonomy. In Pool Party, attributes are fields where you can input information freely. If we go back to our example, we could now add attributes to each of the classes we have defined. We could, for example, create an attribute for beverage that is called image. It could be that you would like to use this field to link your concept to an image of that concept that is stored somewhere else. Since alcoholic beverage is a subclass of beverage, all members of the class alcoholic beverage would also be allowed to have the attribute image. Now we can go ahead and specify an attribute for alcoholic beverage that is called % alcohol per volume. This attribute will only be available for concepts of the class alcoholic beverage. The third thing we define in ontologies are relationships between concepts. You have already heard in previous sessions that in Pool Party you can define certain relationships between concepts. For example, you can say that two concepts are related. Now, in an ontology, you have the opportunity to define additional kinds of relationships. You can be much more precise when you build your thesaurus, because now you can say what kind of relationship two concepts have. Let's go back to our example. In order to establish relations between classes, we first need to think of another class. We could define a class called cocktail. Now we could start creating relations between our beverage or alcoholic beverage classes and the cocktail class. As an example, we could define a relation from cocktail to beverage that we call contains. We could use this to specify which beverages go into which cocktail. Now we would only be able to see which cocktail contains which beverage, but we would not be able to tell which beverage goes into which cocktail. If we want to be able to know this, we would have to specify another relation. We could say that a beverage is contained in a cocktail. Now we would be able to tell which cocktails a beverage goes into as well. What we can also do is create relations between instances of the same class. For example, we could say that cocktails can also be related to each other. For example, we could say that cocktails can be variants of other cocktails. Let's recap. In an ontology, you can define classes, attributes, and relations of concepts. For every class, you can define attributes. Between classes, you can define relations. Now we have heard what an ontology is and taken a look at a small example. Another example.
Okay, so So the ontologies is kind of confusing, to be honest. So the types of classes. So basically you have a subclass. This time we did alcoholic beverage. Of course, don't underage drink. So we have an alcoholic beverage, and that's a subclass to the class beverage, because this is a more specific example of yeah, that's correct there. Okay, so this is a subclass. It's in a relationship with this class. So this is an alcoholic beverage, and this one's a beverage. This is more specified version. So attributes, they basically describe, we call it properties. So properties, we basically describe concepts with additional information. So if you want to specify further. And then relationships, you can precisely specify the kind of relationship between concepts. So right now, it seems it is like a very sophisticated version of like a data finding thing. So it's like a sophisticated version of a thesaurus and all these things. So I want you guys to let me know, what do you think the benefits are of using ontologies? Yeah, it's easier to understand when you look at it because there's quite a bit of data and like explanations that go into these ontologies. So it's pretty good. Yeah, so we can form different relationships with groups, the classes, which can help us understand the world. So basically, what happens is you put in a class, and then it shows the subject that you search, the entity, and that can help us understand quite a bit, which is very important in biological. And the biological ontologies, they're important to connect the data set as well as data integration. We'll get into that later. We can link the process of evolution of the world species. So basically we can combine a bunch of data, and it's very important. It can be important to biological like ontologists. This is a picture I took of the video, and this is basically what it shows. So this is a subclass to the class, the beverage, which contains percent alcohol. This is the attribute, or we call it the property. And that can give us a very precise answer for what we're looking for inside the ontologies. So here we have the NCBI, which is very valuable. So it stands for the National Center for, I'll rip Aaron, let me see. Is he back in? Okay, so now we see the, it stands for National Center for Biotechnology and Information. So it's pretty easy to access. It's open source to many databases, softwares, tools, and it offers course tutorials on how to use tools. I actually, a couple of days ago, I actually watched some of the video and they're very helpful. The GenBank database, it's primarily the use of nucleotide sequence of all the available genes that are publicly available. And then the ref sequence, we can well annotated database, which includes DNA, transcripts and protein amino acid sequences. And then here are all the other tools. So let me go here. It's my screen sharing the same one. All right, so basically we can go to classes, and here are the cellular organisms. we can pick the subclass, let's say bacteria. And that's the subclass of the cellular organisms because it's more specific than we have properties. And then, yeah, it's pretty straightforward, I guess. It's visualization, path, cellular organisms, taxonomy, and class mappings. This is like all the stuff that others have done. So yeah, it's pretty useful for biologists, and not just biologists. Like, let me go to the ontologies. So what we're talking about is biological ontologies, but let's see. So our ontologies, we can use like many different ontologies. Like as you can see, these are all ontologies, and we went through a lot and it's like nothing. So yes, there are a lot. It's very interesting, I'll say. It's a little bit complicated to understand because, you know, science jargon and stuff. It appears that scientists like to make average citizens like very confused like me. So yeah, so that's basically the biological ontologies. And then let me screen share this. And then BLAST, this is for sequence. So there's an algorithm which compares the nucleotide sequences or the amino acid sequences. And this is very important. Like in high school, we did this, and it's a little bit confusing, but it's a useful skill to know. Like, it allows for comparison of your DNA, RNA, or protein sequence to a library of sequences. And then it's a text-based format of nucleotide or peptide sequences, and the output is graphical alignment summaries, table with e-values for various similar sequences. And then here's the family of BLAST programs. They're quite a bit. So the nucleotide BLAST, I'm pretty sure that's the one we use. So given a nucleotide query, it'll return the similar DNA sequences and species from the database specified by the user. Then there's a protein BLAST. This returns the similar structures, structural proteins from the database. The nucleotide 6' translation protein BLASTX. This compares conceptual translation products from DNA sequence to a protein database specified by the user. And then the protein nucleotide 6' translation tBLASTN. So this compares a protein query against all 6 translated frames of nucleotides here. Of course, there are like a lot of BLAST programs, and there are a lot of tools on the NCBI website, but these are some pretty good ones. And then here we can find we can find like Homo sapiens, which are like us. So what we do is we find the we do the blast, and then we find the alignments, and then that'll basically give us the score and identity. And the applications of the blast is like it establishes the phylogeny and creation of phylogenetic trees. And for researchers, finding a similar gene to knock out in a test organism to determine a gene function, that's very important, because when you want to like knock out in a test organism or find out a gene function, it better be accurate, because if you mess up, then I mean, you'll find the wrong gene function, of course. And when you test an organism, you want to be able to find good data and a good test to see if we can apply that to humans or other animals. And then it can locate domains in your protein sequence, active site. Well, okay. And then now let's talk about data integration. So first of all, do you guys know what data integration is? I know I'm throwing a bunch of new vocabulary here, and it's a bit challenging, but I want your best guesses. Okay, so basically data integration is the process of combining data from different sources into a unified view. So basically what that means is we can find different data points from different sources and stuff to one single unified source, which is very, like, this is important stuff, because when you combine the data, that'll tell you a lot more than the data separately. So yeah, it can combine and research results from different bioinformatics repositories. And then here's an example. So large scale science requires different data sets, so the data integration can combine the different data sets. So let's say, I don't know, okay, so there's a number of species, and then they sampled it from different areas. And then these are different, so they sampled them, they're different data sets. We can combine these data sets, and that'll be a much better sample size or use. Oh, I didn't get to see the chat. Let me see this. All right, good answers, guys. So it's the yeah. So this is a very important tool, because in the sampling point, you want to have very good samples. This will lead to better data and more accurate data. And basically, that's what we want. Because if we have inaccurate data, we can't make very good predictions, or we can't do very good current tests and stuff, which is That's not good. Okay, so how do biological ontologies impact data integration? So these kind of go hand in hand together. So biological ontologies and data integration help with access and analysis through their use in capturing and structuring biological data. So this will enable computational reasoning over semantic systems. And ontology-based data integration involves the use of ontologies to effectively combine data or information from multiple sources. So with the use of both biological ontologies and data integration, we can access and analyze more accurate data, which is very important. And this will help us make better predictions about like the environment with biodiversity informatics, whatever we're looking for on the ontologies. The data integration will help quite a bit with the data. All right, and then I'll hand it off to Chris and
Cool. Can everyone see my screen? Oh, wait, I have to open the chat.
yeah.
Okay. So I'll present now. Hopefully this will work. Joanne will talk about the scientific method a bit.
Okay, so before I go into like more real applications of bioinformatics and biological ontologies, does everyone sort of understand or at least have an idea of what bioinformatics and ontologies are, just so we're all on the same page? Okay, that's good. So if you're like a student who wants to do research, you'll first start off by asking yourself, what problem do you want to solve? Or narrow down your focus, like ask yourself a question. For example, say you're interested in, or like you read the news a lot on new biological discoveries, and then you become interested in Parkinson's disease, for example, then you start to do background research and read more on Parkinson's disease. And then you might find like, oh, Parkinson's disease has symptoms like irregular motor activity. And then that links to problems in the brain. And then from there, you start to narrow down, oh, there are these things that haven't been studied yet. And these things could be improved in treatments, diagnosis of Parkinson's, things like that. And from there, you determine where to start your research and you construct a hypothesis. For example, if I'm using the Parkinson's example, we'll also provide you a better walkthrough of a different example, but say you want to focus on a gene directly related to Parkinson's. I think one is PARK2. You would probably go to you would probably read a lot of articles on that gene, and then maybe you read some articles that seem like they contradict each other. Like one says this gene is involved in protein binding, and another says the gene is part of this complex in the cells in the brain. And that's when you might go to a biological ontology and search that gene up. And then from there you would see, oh, there are all these sort of different definitions of the PARC2 gene, it has a role in protein binding sites, but also it's part of this important complex in your neurons. And then say, where does bioinformatics come to here? Where do you find data and what do you do with it? There are likely a lot of published articles and research papers already on that specific gene, and you go to there and you'll see a lot of data that scientists have published with research done on that gene, and then you would see the sort of experiments they've done, as well as use the experimental data in their research. And you can kind of see in the image on the slide how that how your research will develop. And that's a basic overview of how bioinformatics and ontologies and all these sort of things that Nathan introduced earlier will be incorporated in real life research.
Yeah, and hopefully you guys can hear me. Just to add on to that, obviously like the scientific method is not like foolproof. There's obviously gonna be mistakes that can be made along the way. And a lot of times you'll find yourself going back to like your hypothesis, modifying your hypothesis, retesting your experiment. And in the scientific community, I know that it's really hard, it's really easy to get confused with all these fancy scientific words and like jargon, but most scientists, if you present them with something like, for example, like Joanne was talking about Parkinson's and like PARC2, there's not a lot of scientists or like the general group of scientists that know like everything about PARC2. It's just specialization. So don't be too like scared by the word choice, because not all scientists will know everything about everything. So just as on, like when you're gonna do a research project, you'll probably specialize in one sort of area. And it's similar for a lot of other scientists. So we're gonna work through an example. If you guys could work through this with us on your computer, that would also be great. We're gonna be using like, we're gonna be showing you some examples of gene ontology programs. So the gene that I chose was ApoE4. So the ApoE gene is heavily involved in Alzheimer's disease. I'm sure that a lot of you guys have maybe not have had firsthand experience with Alzheimer's, but maybe know a few people who have. And the ApoE4 allele specifically is thought to be involved in Alzheimer's. So we are just going to be looking at a few ways we can learn more about this gene. And if you guys want to follow along, you guys can use the ApoE4 allele, or you can just use any gene that you want. You can search up like any like disease that you're interested in, like maybe a muscular dystrophy or like sickle cell anemia. And then if you search it up like on Google, you might find a gene associated with it, and you guys can do a similar thing. And if you do that, just type in the chat what gene that you wanted to test. And tell me if I'm going too fast or slow as well. So right now we're just going to be looking into Alzheimer's disease, and like obviously a lot of us can't do like wet lab work. We can't really be in a scientific lab right now, especially because of the pandemic. So what we can do is use these computational tools like the software that's already been developed, and we can look into how there's different scientific mathematical correlations. And that's possible to do at home. So first, we'll be talking about gene cards. If you guys can right now search up gene cards on your devices and just type in the chat like, I don't know, that you've found it. It should look something like this. But while you're doing that, I'll just, Gene Cards is basically, yeah. So Gene Cards is basically just like, it's a software. It's like a website that gives you a lot of relevant information about a certain gene that you're looking into. So it's not really like the program like BLAST, like Nathan talked about earlier, where you would actually be physically manipulating data and like inputting your own sequence to find output sequences and organisms and such. But Gene Cards is kind of like a big like Google for genes. So it's really helpful. Like if you've looked into the gene, like Joanne's example, PARC2, you can search up PARC2 here, and then you'll get to know a bit more about that and what it's involved in. So right here, I searched up I just call it ApoE. That might not be the way to pronounce it. Yeah, and if you guys do the same thing, you guys can explore any gene that you want. I'll give you a hint, like a lot of genes, there's a lot of genes in our body, like there's like 20,000 or so. So if you just type in four letters, there's a chance it might be a gene. And then once you click search, you get a bunch of results. So like if I search up ApoE4, all of these different genes come up. So like ApoE, APP, PSEN1, those three are very heavily thought to be involved in Alzheimer's. PSEN1 a bit less so, but APP is the gene that creates the protein that like clumps together and basically blocks a lot of these brain cells in Alzheimer's. So as you can see, if you search this up, you'll get a lot of correlating genes. Like they would be, like Nathan said, like in the similar class. So they would be in the same class of like genes that may have attributes pertaining to Alzheimer's. And then, so hopefully you guys have like searched something up, any gene, and then you get a bunch of information. So you get like different names that it goes by. This is why, like again, scientific jargon, like there's so many different names for ApoE4. So scientists like to be confusing. There's like scientific disorders, like probably if you click on this, Alzheimer's will come up, or like other neurological disorders. It will tell you like even where you can buy certain products, like certain drugs that are already out, animal models, publications about this gene. So definitely I recommend, like if you're just looking into a gene and you don't know that much about it, Gene Cards is a good place to start. Yeah, and make sure you type in any questions if you have any questions. But now we'll be talking about GEO, which stands for Gene Expression Omnibus, and it's another like ontology software. This is it's similar to BLAST. It's on the NCBI website, and it's a bit more hands-on where you can actually manipulate the data yourself. Like Gene Cards is more of just like a big Google for genes, and GEO is like, I'm bad at analogies, but GEO basically you get to actually tweak the data a bit. So like in a study that you're reading, like a few weeks ago, I believe I was reading a study about pesticide degradation, and a lot of studies will cite like a GenBank database. So they basically, once they're doing a study and they have all this data that they found, like they found DNA sequences of a certain like bacteria, they'll be inputting it into a database so other scientists can also use it and analyze it. So, and then they'll have an accession number in their study. And then if I searched up that accession number in GenBank, in GEO, so if you guys want to do it right now, you don't have to, because it might be a bit fast, but like if you go to GEO and you search up a gene that you want, then you'll also get a lot of studies, but I'll show you that a bit later. So GEO accession numbers, you can enter the GEO accession number. There's different ways to analyze a sequence with different bioinformatics software. And then here is another study that I might get back to. Yeah, I might get back to this slide. So GO2R is basically more of it's a better way to like analyze the data instead of just looking at raw data sets. So a research topic I'm interested in is looking at nicotine, and nicotine may have therapeutic benefits in people with brain disorders. I'm not telling any of you young kids to ever be exposed to nicotine, but there's a certain body of research that we're looking into right now. So if I search up nicotine in Geo, then like if you press the search box, you're gonna get like this little box that pops up that says like there's this many results for in the Geo datasets database, and there's this many results in the Geo profiles. I click the first one, the datasets database, and then you get a bunch of like different sequences, or I mean, sorry, you get a bunch of different studies or publications that have inputted data here. So you can look through like your results. I clicked on the first one, but the first one wasn't available to analyze with GO2R. So just keep looking, find something that interests you. As you can see here, there's organisms that come up. So I want to do it, I want to look at it in humans, not other organisms, because humans are the most accurate to study about humans. So I would choose a study with Homo sapiens, not another organism. And then I found one here that's like, if you click on the link, it will take you to this page. It will give you a short overview of what the researchers did. It will tell you the number of samples. So the sample size is really important, because when you're doing an experiment, obviously there can be errors in your experiment. So you want a lot of samples to make sure your data is actually probably more accurate. So here it has like 221 samples. I think that's a pretty good number. If you like, try not to choose experiments with like 1 or 2 samples. Those probably won't be significant at all. And then I just clicked Analyze with GO2R. That takes me to this page here, which I'll see if so here. This is the, hopefully you guys are still seeing my screen. This is the GO2R page. And then from here, you can define groups. So like, as you can see, this one's looking at like the nucleus accumbens, which is like a region in the brain that deals with dopamine of smokers and non-smokers. So it's kind of seeing how nicotine like affects, and other compounds affect like your dopamine production. So you can click on groups and then set a certain group. So here, like if I say define groups, I have a smoker group, so I can add it here. So I can select these samples and add it to my smoker group. I can create groups just by adding a name. So I could like, if I enter that group, I can add more groups here. So you can add up to 10 groups in GO2R. But I feel like for this data set, since I want to see the correlation between like smoking and not smoking, I would create 2 groups, one for smokers and one for non-smokers. Hopefully this makes sense, guys. Please let me know if it doesn't. And you might have a bit more experience with this later on in the camp. We can definitely walk you through it because this is going a bit fast. And then here, you can use like the researchers' study subjects that they inputted. A lot of these researchers will input genes associated with like their study subjects. In this example, they're flies. And then there's different ways you can analyze it. So this software is just really good for showing differential gene expression, or different ways genes are expressed in different subjects. So for example, in this study, I made a group called control where they weren't exposed to nicotine, and then an exposed group where they were exposed to nicotine. And as you can see here, for, which gene is this? For the HR38 gene, I'll get my pointer. For the HR38 gene here, like this gene was expressed a lot more in the exposed group than the control group. So it tells me that nicotine has an effect on this gene. So there's different ways you can do this using like different softwares. And there's also something called value distribution that just tells you how well suited your data is. So this is just an overview. Like the main point I'm just trying to get across is you can use these different softwares to see how different genes work. So yeah, and now we have a Kahoot for you guys. Hopefully this made a bit of sense. If you guys want to try it out later, that would be good as well.